Hello, and thanks to everyone for participating in this webinar on an introduction to the Multimodal Animal Rotation System from Chemistry Molecular Imaging. My name is Gilbert Feek, and I'd like to start by discussing the differences between single-view planar versus multiple-view rotational imaging. Traditional single-view in vivo imaging can be challenging for fluorescence and luminescence modalities due to the depth-dependent attenuation of light in tissue. As you can see in this cartoon, critical molecular signals emitted from the far side of the animal could be completely missed by the imaging system. Even signals which are visible from a particular view may not be optimized due to the animal position being slightly suboptimal. For example, ideally the molecular signal shown here would be aimed straight at the camera via the shortest path through the tissue, instead of having to cross a thicker path through the tissue as shown here, resulting in less brightness. Now, I should point out that X-ray imaging doesn't suffer from tissue-dependent problems like optical imaging. However, observation of endogenous or exogenous X-ray contrasts may be similarly suboptimal if the choice of animal angle causes obscuration of features. Multiple-view in vivo rotational imaging, on the other hand, provides the best opportunity to find and optimize molecular signals. Here we see the animal rotating around its craniocaudal axis. At each angle it stops and an image is captured. With sufficient angular resolution, any observable molecular signal can be found and optimized. So comparing images across longitudinal studies for multiple animals is safeguarded by scanning for the optimal signal, and potentially fewer animals could be used to obtain a target statistical significance because each animal is examined more carefully. Also, X-ray imaging can benefit from the rotational approach to find the best angle to view the animal. Chemistry Molecular Imaging recently commercialized the MARS, which stands for Multimodal Animal Rotation System, and includes hardware and software to automatically acquire a series of multimodal images of a mouse from multiple angles, as much as a seamless 360-degree scan, in order to capture light emitting from as many tissue structures as possible. The new hardware includes a rotator insert and add-on controller module, which is compatible with the In Vivo FX Pro, Multispectral FX Pro, and DXS Pro systems available from Carestream Molecular Imaging. The new software includes new features in the Carestream MI control imaging software, such as rotation controls in the preview and protocol dialogues, and the Carestream rotation visualization software that lets users browse images by angle, view animated playback, and export rotation movies. So how does the MARS work? The rotation module is a motorized insert that installs into the imaging chamber. Here's a close-up view. Loading the mouse is quick and easy. Just open the lid and drop the mouse straight in. The MARS accommodates one anesthetized mouse having a wide range of size and weights. Anesthesia, warm air, and exhaust lines are integrated into the unit. The mouse is automatically rotated by the torsional support film to the desired angles for sequential multimodal imaging, which is either on command for preview, single image capture, or is rotated according to a preset imaging protocol for single or multiple angle capture. At each angle, one or more images using one or more modalities is captured. When the image capture is complete for a given angle, the animal is rotated to the next angle and the process is repeated. The entire set of image data are saved to disk. The image data are available for standard image analysis and presentation in Carestream MI or in third-party software such as ImageJ and Adobe Photoshop. The image data are also available for advanced angle index browsing and animated playback using Carestream rotation visualization software. It takes one click to open the desired image in Carestream MI for analysis. Also, the dataset can be exported as an AVI movie for presentation. The MARS is compatible with all modalities available from Carestream in vivo on the Carestream in vivo imaging system, including fluorescence, luminescence, radiographic, and radioisotopic. The MARS has the potential to enhance any application where multiple view examination of the animal is advantageous. For example, cancer cells, stem cells, inflammatory disease, etc. Here's one example where the Mars was used in a tumorigenesis application to search and optimize signals from subcutaneous, intraperitoneal, and secondary tumors from ovarian cancer stem-like cell xenografts, which were pre-labeled with near-infrared fluorescent nanoparticles. Tumorigenesis was monitored across longitudinal studies. This slide shows the fluorescence images and corresponding X-ray anatomical overlays over different angles at one time point in a longitudinal study. The graphs at the bottom show the brightness of 3 tumors as a function of angle. Without multiple view rotational imaging, critical features may be missed entirely. Even when features are visible from a particular view, they may be suboptimal. With rotational imaging, the final result from an imaging session can simply be the maximum signal from the feature across the rotational data set, which can be tracked across a longitudinal study and/or multiple mice. The rotational imaging guarantees that any observable signal can be optimized, which can serve to reduce noise in the experimental data set and potentially mean that fewer animals would be needed to achieve a target statistical significance. And this slide shows the ex vivo validation of the intraperitoneal tumors that were found during rotational imaging. Here's another example showing how the detection of small populations of fluorescent cells is optimized by rotational imaging. These were cells labeled with near-infrared fluorescent nanoparticles, and a serial dilution was injected subcutaneously at 3 separate sites. As the cell population size decreases, the angular window for detecting the cells also decreases. Here's another example pertinent to X-ray angiography. A barium sulfate vascular contrast agent was administered via carotid artery cannulation using a syringe pump prior to post-mortem rotational imaging. The precise angle selection that Mars provides has the potential to minimize standard deviation of vascular density measurements comparisons across cohorts of animals. Rotational imaging minimizes the need for X-ray analysis of extracted organs and tumors. This slide shows a survey of fluorescently labeled organs. The fluorescent signals depend on the craniocaudal rotation angle. The optimal rotation angles for the stomach, intestine, kidneys, and bladder are reproducible among multiple mice. In summary, the Multimodal Animal Rotation System, or MARS, from Kirschner Molecular Imaging is a unique tool that relieves concerns about missing critical molecular signals in small animals, provides a wealth of data for signal optimization and precise comparisons across multiple animals in longitudinal studies, and delivers visually appealing animated presentations. This concludes this webinar, so thank you.